Hey, welcome to today's session. I had a lot of questions yesterday regarding transporting the samples for DNA barcoding. So we thought we will make a small video for transporting the samples. So I'll be talking about the preservatives and how you can transport three different samples. Okay, microbial samples, which includes bacteria, fungi, and viruses, and animal tissues, how to transport them, including insects and all kind of animal tissues, plant tissues, including whatever part, but usually we accept the leaf. We will see them one by one in this video. Okay, so animal tissues, oh no, sorry, microbial tissues. First of all, we'll start with microbial biomass. So we have bacteria, we have fungi, and we have phages. Whenever I say viruses, I mean phages. We are not equipped to handle BSL 3 and 4 category. We can handle BSL 1 and 2. So bacteriophages, whenever you see a blocks, you can take an agar button, store it in a sterile distilled water. You can send us, or you can simply send the plate like this. We will take care of list. Okay. Fungi, yes, we do accept plastic plates or glass plates where you can have a fungal pure cultures. You can send the fungal pure cultures. We are adapting CTAB method for extracting DNA, and we will go for ITS gene amplification and go for sequencing. Okay, and for bacteria, a pure culture in quadrant streak, if you can provide, which has isolated colony in the last quadrant, we can easily go ahead with your live bacterial sample. In barcode, I guess they are accepting only live bacterial and fungal because many are doing microbiology when they are doing for first time. Quarren sticking itself is a big challenge for them, and many times I'm seeing bacillus colonies, translucent colonies around the any pigmented colonies are around the target colonies, where a professional microbiologist can easily distinguish, but amateurs they may not. Okay, if you have extracted DNA and done the PCR from such a mixed culture, you may get a precise 1500 base pair in your PCR amplicon, but during the sequencing, you will have a fuzzy chromatogram. So, which is impossible. Then it complicates the process of resequencing, paying the money again. Customers, there is a fight between the sequencing department. To avoid all these commotions, we came up with this idea that we will decide the purity by inspecting the pure cultures, and then we will go for DNA isolation, PCR, and sequencing. So this is for microbial samples. And here we have one for animal tissues. Okay, I would suggest just 100 milligrams of sample in zero point 0.5 ml of 70% ethanol will do. Animal tissue, we can do that. Okay, no need to preserve the entire animal or a big insects. No need. Just leg parts of insects cut down into multiple pieces, stored in either 2 ml Eppendorf tubes or 1.5 ml Eppendorf tubes, or in case of very big animal, 15 ml or 20 ml. But I'm still recommending. See here, I have a few insects here in 5 ml or 2 ml tubes. This is much more enough to send the sample for barcoding. Okay, so why ethanol and why 70%? Ethanol can instantly dehydrate the entire tissue and remove the moisture from the tissue. They prevent the tissue from autolysis of their own enzymes. So they have multiple uses. They also prevent the tissues from microbial degradation. So why 70%? Why not 100%? When you use 100% ethanol, the preservation action of the ethanol is very instinctive, and many times the cell actually becomes very hard in 100% ethanol because of dehydration process, and the ethanol penetration into the tissue is actually reduced. When you have a 70%, the penetration is better, 30% water. Okay, so they have to get into each and every cell in the animal so that they can preserve better. Their enzymes and lipids has to be fixed, precipitated, or coagulated so that they don't disturb their own DNA. Okay, that's the idea behind using 70% ethanol. All right, and also it prevents microbial degradation. Okay, so facilitate that if you some animal may have more protein or more lipid in their biomass. If you are immersing your tissue in the ethanol, if you are seeing your ethanol is instantly changing color, give it some 2 3 hours time. If the ethanol is completely turbid, I would recommend just pour off that ethanol and add more fresh ethanol, fresh 70% ethanol. Okay, no need to store it in 4 degree or ice cold temperature. Just send us the transparent ethanol preserved tissue samples. We can take care of it. Okay. So changing ethanol might be required in some cases. Even in many times, I have extracted DNA from that colorized ethanol. The first ethanol I'm going to change, instead of throwing it out, I can simply keep it in 65 degree, 70-degree water bath. Let the ethanol evaporate. I will add lysis buffer to the residues, and I can extract DNA from that. Okay. So that's how the animal tissue should be preserved and transported. And when it comes to the plant tissue, I would suggest same ethanol. Okay, I know many people will be surprised. See, this is best. We have seen this is working for DNA barcoding in plants. Okay, cut them into little pieces so that ethanol penetrates into every tissue better. And like I said, for animal tissue, if you are seeing ethanol decolorizing immediately, maybe change the ethanol after some time. Okay. Yes, ethanol is not recommended for long-term storage. For short-term, it is okay. Many high quality DNA, high molecular weight DNA can be isolated from such a preserved tissues for 3 days time. Okay. If you want to preserve it for 5 days or 7 days, maybe put it in 4 degree, it can be done. Okay. You can preserve the tissues in the ethanol and do 4 degree. There will be a degradation, but still remember, we are not going to target nuclear genome of the plant. The barcode genes, RBCL and MATK for plants are actually chloroplast genes. And their circular genome, they are much more stable than nuclear genome. So in my estimate, even I have received the samples from Mizoram and other northeast parts of India, which took 4 days to come. They are preserved in ethanol. Yes, when it is 4 days, no ice, there could be a degradation, but we don't care about nuclear DNA degradation because our chloroplast genes, they are actually multiple copies in number, and they are much more compact than the nuclear, so they resist degradation. My estimate, I would say, ethanol preserved leaves for barcoding can be used up to 9 days at room temperature, up to 9 days. So whenever we receive the sample within India, we don't, if it is, you don't need to preserve in the ethanol for first 3 days. Simply send the leaf. If you are going to process on the same day of collection, no need to preserve in any preservative. If you are around southern part of India, I would say just take the leaf, put it, and we can courier it to us. If you are sending from Mizoram, maybe 3 days, 4 days it will take. Just put it in ethanol and watch for 3 hours. If the color changes very rapidly. Pour off the ethanol and fill it with the refresh. Top it up with the fresh ethanol and then send us the courier. It is working better and it is working fine. Okay. So these are the ways you can preserve the samples and send it to us. Thanks for watching.